Welcome to our deep dive into just a really fascinating review article from a 2026 issue of Nature Biotechnology.
Yeah, thanks for having me. It's a great paper.
Our mission today is to explore how the massive discovery power of mass spectrometry is, teaming up with wearable biosensors to basically completely change how we track human health.
Right. It's a huge shift.
And to start, I want you, the listener, to just imagine for a second that your doctor only ever gets to see a single blurry photograph of you, like once a year.
That's a highly unsettling thought, honestly.
Right. They look at this static delayed snapshot, and from that one image, they have to figure out literally everything about your health. How you handle stress, what your diet is doing to your cells, whether a hidden infection is brewing.
Obviously, you can't see any of that in a single photo.
Exactly. It sounds absurd, but realistically, that is exactly how modern molecular diagnostics work today. Okay, let's unpack this. Why is relying on that static snapshot actually dangerous?
Well, the primary danger is the time gap. I mean, whenever we rely on invasive sampling, like a standard blood draw, it demands this whole clinical infrastructure.
Right, you have to actually go somewhere.
Exactly. You physically travel to a clinic, a phlebotomist draws the blood, the vials are shipped to some centralized lab, and then the analysis happens days later.
Which is agonizing if you're waiting for results.
Yeah, and for a chronic, slow-moving condition, maybe that delay is, acceptable. But for acute conditions, things like sepsis, a severe respiratory infection, or a cardiac event, meaningful physiological changes happen in a matter of minutes.
Minutes, wow.
Yeah, the molecular signals that your body is in severe distress are flashing red, but the current medical system simply isn't designed to look at them in time.
So we have a system built on delayed blood draws, which brings up an obvious question. If blood is too slow and honestly too painful to constantly sample, where else are we supposed to look?
And that is where the biofluids come in.
Right. The source material details this massive menu of what it calls non-invasive and minimally invasive biofluids. But I have to admit, looking at this list, I am highly skeptical.
Oh, why is that?
Well, it mentions sweat, tears, saliva, the moisture in our breath. It even lists cerumen, which, if I'm not mistaken, is just the medical term for earwax.
It is indeed earwax.
Yeah, I mean, I understand these are easy to get, but how can sweat or earwax possibly be as informative as blood? It sounds a little gross, to be honest.
I get that. Might sound unconventional or gross, but these fluids are absolute gold mines of real-time biological data. The key is understanding what we are actually looking for.
Okay. What are we looking for?
Well, a lot of people hear the word diagnostics and they immediately think of DNA testing, but your DNA is just a genetic blueprint.
Right, it doesn't change.
Exactly. It tells us your inherited potential, but it is static. It doesn't tell us what is happening to you right this very second. The biofluids you just mentioned, however, are packed with metabolites, lipids, and proteins. We call these downstream effectors.
Downstream effectors.
Okay, I see. So if doing a DNA test is like looking at a factory's architectural blueprints to figure out what they might build, looking at these downstream effectors is like walking onto the factory floor and looking at the actual products and the exhaust rolling off the assembly line right now.
That is a very accurate way to visualize it, yeah.
Yeah.
These molecules capture the rapid functional responses to stress or disease.
So they show the work in progress.
Right. And different fluids tell us different stories about the body. Take interstitial fluid or ISF.
What is that exactly?
This is the fluid sitting in the microscopic spaces right between your cells. We can access it using tiny, painless microneedles that barely scratch the surface of the skin.
Oh, wow. So you don't even feel it.
Exactly. And ISF directly mirrors the capillary exchange from your blood, which makes it an incredible proxy for tracking systemic markers like glucose or lactate.
Okay, so ISF is like a window into the blood without the needle pain. What about sweat? Sweat, conversely, gives us a highly localized window into thermoregulation, electrolyte balance, and your nervous system's activity by revealing stress hormones like cortisol.
Okay, let's pause for a second to do a quick recap and make sure we're seeing the big picture here, because we're covering a lot of ground.
Sure, sounds good.
So, bullet point 1, blood tests are too slow for emergencies. Bullet point 2, our bodies are constantly leaking live streaming data through alternative biofluids like sweat, tears, and ISF. And bullet point 3. These fluids contain downstream effectors, which are the real-time proof of what our cells are actually doing.
That is a perfect summary.
Which makes me wonder, for anyone listening right now, if your body is constantly broadcasting its status through your sweat and breath, What specific stress or health signals do you think you're radiating at this exact moment?
It is wild to think about.
It really is, but knowing those signals are there in a drop of sweat is one thing. How do we actually read them? The paper talks about something called mass spectrometry, or MS. What is this machine actually doing?
Mass spectrometry is essentially the ultimate molecular decoding machine. The paper refers to it as an untargeted omics platform.
Untargeted omics. That sounds complicated.
It sounds intimidating, but to understand why it matters, consider how current diagnostics work. Usually, they are targeted. A doctor suspects you have a specific condition, so they order a test for one specific biomarker.
Right, like checking my cholesterol.
Exactly. You have to know what you are looking for in advance. Mass spectrometry flips that entirely. It maps thousands of molecular features simultaneously without any prior hypothesis.
So it's like casting a massive high-tech net into the ocean to see every single species of fish that happens to be swimming by, rather than sitting there with one specific fishing lure hoping a single trout bites.
Yes, that's exactly it.
But how does it actually identify thousands of microscopic molecules at once? Walk us through the physical mechanism here, because the paper mentions lasers and magnetic fields, and honestly, it sounds like science fiction.
It's an elegant piece of physics, really. At its core, mass spectrometry does three things. It gives molecules a static electric charge, it accelerates them, and then it weighs them.
Wait, it literally weighs individual molecules.
It does. Imagine taking a sample of your sweat. First, the machine ionizes the sample, meaning it blasts the molecules so they gain an electrical charge.
Like static electricity.
Basically, yeah. Once they are charged ions, the machine shoots them through a vacuum chamber using an electric field. As they fly through this chamber, a powerful magnet bends their path.
Ah, okay. So it's like throwing a baseball and a bowling ball through strong crosswind. The lighter baseball is going to get blown way off course, but the heavy bowling ball will resist the wind and stay mostly straight.
That is the exact principle. By measuring exactly how much each molecule's path curves in the magnetic field, the mass spectrometer can calculate the precise mass and weight of every single molecule in that drop of sweat.
That is mind-blowing.
It is. And because every protein and metabolite has a unique known mass, we can identify exactly what is in the fluid.
But fluids are also different. I mean, sweat is a liquid, breath is a gas. How does one machine handle all of those physical states? The paper uses all these acronyms like LC-MS, GC-MS.
Right, so the core weighing mechanism, the mass spectrometer itself, stays the same. What changes is how we feed the sample into the machine.
Okay, so what does LC-MS stand for?
LC-MS stands for liquid chromatography mass spectrometry. The chromatography part just means we run the liquid sweat or saliva through a special column to separate the molecules slightly before we shoot them into the mass spectrometer.
Got it. And what is that good for?
This is fantastic for proteins and small molecules. In fact, one study cited in the review used LC-MS to analyze sweat and discovered a specific 5-metabolite panel that can differentiate lung cancer patients from heavy smokers with roughly 80% accuracy.
80% accuracy just from a swab of sweat. That is staggering. And I imagine GC-MS, gas chromatography, is how they handle the breath samples.
Yes, exactly. GC-MS is ideal for volatile compounds, molecules that naturally evaporate into a gas. Researchers use this setup to capture over 100 specific chemical compounds in human breath that are actually predictive of acute cardiovascular disease.
The one that really caught my eye was MALDI-MS laser desorption ionization. It sounds like they're literally shooting lasers at bodily fluids.
They actually are. MALDI uses highly focused laser pulses to instantly vaporize and ionize a sample. It is incredibly fast and solves a major physical problem, which is sample volume.
Because they only have tiny amounts of fluid to work with.
Right. When scientists use engineered nanomaterials like gold or silver nanoparticles to help absorb the laser's energy, they can analyze vanishingly small amounts of fluid. Using this technique, they detected bladder cancer signatures from just 0.3 microliters of urine.
0.3 microliters.
Yeah. They even identified Parkinson's disease signatures from a mere 10 nanoliters of saliva.
10 nanoliters. That is a fraction of a drop. But wait, that tiny volume brings up a massive constraint I saw in the text. Human skin is covered in bacteria.
It is. We are covered in them.
We have a whole microbiome living on us. So if I give the machine a fraction of a drop of sweat, how does the mass spectrometer know if the protein it just weighed came from my human cells or from the Staphylococcus bacteria living on my arm?
That is the hurdle of sample purity. and it requires some serious computational heavy lifting known as metaproteomics.
Metaproteomics. Okay, break that down for us.
When the mass spectrometer weighs all those molecules, it generates a massive data profile of everything in the sample. Instead of just comparing that data against the dictionary of known human proteins, the software uses concatenated databases.
Meaning it mixes dictionaries together.
Essentially, yes. It combines the human genome dictionary with the bacterial microbiome dictionary. Advanced algorithms analyze the exact structure of the protein fragments and assign them taxonomically. It mathematically untangles the human biology from the bacterial noise.
That is incredible. And it's important to note that the machine isn't just looking for one single red flag, right? Because biology is messy. If my lactate levels are high, that could mean I have a life-threatening blood infection, or it could just mean I just printed up a flight of stairs.
Right. Single biomarkers are notoriously unreliable. That is why MS looks for what the authors call multi-analyte fingerprints and pathway-constrained ratios.
Give us an example of that.
Well, instead of looking at lactate in a vacuum, the machine calculates the ratio of lactate to pyruvate. This specific ratio reveals the actual cellular energy state and the oxygen utilization inside your cells. It cuts through the environmental noise and gives us a true physiological diagnostic signal.
Okay, time for another quick 5-minute recap. So we've learned that mass spectrometry is this incredible discovery engine that uses electrical charges and magnetic fields to literally weigh thousands of molecules at once.
Right.
It adapts to liquids, gases, and microscopic volumes using lasers and nanoparticles. And it uses complex algorithms, like metaproteomics, to filter out bacterial interference and find true chemical ratios.
Perfect summary.
It really makes you wonder, to the listener out there, if a machine can map thousands of molecules in a single drop of your saliva, what hidden health patterns might it find that you are completely unaware of right now?
There's probably a lot we don't know about our own baseline health.
Absolutely. But as amazing as mass spec is, there's a giant elephant in the room here. These machines are the size of a refrigerator.
At least, sometimes bigger.
Right. They cost hundreds of thousands of dollars, and they require a team of PhDs to operate in a climate-controlled lab. We started this deep dive wanting a live streaming movie of our health, but you can't strap a mass spectrometer to your arm.
No, you cannot. They are discovery engines, not continuous monitors. The MS tells us exactly what molecules to look for. The next step is translating that laboratory discovery into a wearable device that can track those specific molecules on your body in real time while you go about your day.
Which means we need to talk about the engineering of wearable biosensors, taking rigid, fragile silicon computer chips and making them mechanically compliant with soft, squishy, stretching human skin. How are engineers actually doing that without the circuits just snapping in half the second you bend your elbow?
It is a profound material science challenge. You have to bridge the gap between rigid electronics and biological tissue. Engineers are solving this using stretchable elastomers like polydimethylsiloxane.
Which is what, exactly?
It is a highly flexible silicone polymer, very stretchy. They are also embedding electronics into soft, water-rich hydrogels that mimic the actual texture of human tissue.
The material that blew my mind in the paper was the liquid metal.
Oh, the eutectic gallium indium.
Yes. Because it stays liquid at room temperature, they inject it into microscopic rubbery channels. So when your skin stretches, the liquid metal just flows and elongates inside the channel, maintaining the electrical circuit without ever breaking. It's brilliant.
It really is a marvel of engineering.
Well, once we have a stretchy bandage, how does the sensor inside it actually detect a microscopic stress hormone?
The paper highlights two primary sensing mechanisms, electrochemical and optical. Electrochemical sensors are the workhorses here. They use synthetic enzymes or aptamers.
Aptamers. Okay, we need an analogy for that one.
Think of an aptamer as a microscopic, custom-designed catcher's mitt made out of nucleic acids. This mitt is folded into a highly specific 3D shape that will only catch one target molecule, like cortisol.
Just cortisol, nothing else fits.
Right. When a cortisol molecule floats by in your sweat and lands in the mitt, the aptamer physically changes shape. This physical movement forces a tiny electron to transfer across the sensor.
So it creates a microscopic electrical spark.
Yes. The sensor reads that microscopic electrical current, and the stronger the current, the more cortisol is present.
And what about the optical sensors?
Optical sensors operate differently. They rely on light. The chemical reaction between the biomarker and the sensor creates a visible color change or emits a fluorescent glow that a tiny camera or optical reader can quantify.
That is so cool. And the prototypes they are already building are wild. They mention a strosomic patch.
Yes, the strosomic patch is fascinating.
It sits on your skin, uses tiny microfluidic channels to pull in your sweat, and simultaneously tracks cortisol, epinephrine, and norepinephrine. It is literally measuring the exact activity of your sympathetic nervous system in real time.
Exactly.
But here's where it gets really interesting and where I see a massive logical flaw in all of this.
Oh, what's the flaw?
Well, if I go for a run in the middle of summer, I am going to sweat a ton. But if I sit on my couch in the winter, I might only sweat a tiny bit. Won't a massive volume of sweat just dilute the biomarker? Doesn't that completely skew the wearables data?
You have just identified the single biggest confounding factor in wearable diagnostics. The authors call the solution secretion-aware normalization.
Secretion-aware normalization.
Okay, and the math behind why it's necessary is staggering. Roughly 73% of the total variance in sweat metabolomics is caused solely by the rate of fluid secretion.
73%. So the dilution effect is stronger than the actual biological signal most of the time.
Exactly. If you do not account for how fast the person is sweating, your biomarker data is effectively useless. Therefore, a viable wearable cannot simply measure the target biomarker. It must simultaneously measure the fluid dynamics of the sweat itself.
How does it do that?
Modern wearables integrate concurrent fluid flow sensors, or they track surrogate markers like osmolality, which is essentially measuring how salty the sweat is. By constantly tracking the flow rate and the dilution, the wearable's onboard microchip mathematically normalizes the data.
So it fixes the math instantly.
Right. It cancels out the dilution effect on the fly, calculating the true, accurate concentration of the hormone, regardless of whether you are running a marathon or just sleeping.
Okay, let's synthesize this leap from the lab to the skin. We're taking the specific targets found by the massive mass spec machines, and we are hunting for them using liquid metal circuits and microscopic catcher's mitts that create electrical sparks when they catch a hormone.
Yep.
And to make sure the data is accurate, the patch constantly measures your sweat rate to mathematically cancel out any dilution. It really grounds this science fiction into reality. So ask yourself, if you had a soft, invisible patch on your arm right now, mapping your stress hormones straight to your phone, how would seeing that real-time data change the decisions you make today?
It would probably change a lot of behaviors, yeah.
That behavioral feedback is powerful. But the paper points out that the true breakthrough isn't just a human looking at an app. The ultimate goal is a bidirectional, closed-loop system where the mass spectrometer in the lab and the wearable on your arm actually talk back and forth to each other.
Yes, the concept of fusion analytics. What's fascinating here is that this is where the whole paradigm shifts. In this loop, the mass spectrometer acts as the architect.
The architect, I like that.
It defines the exact physiological concentrations of the biomarkers in healthy and diseased states, which sets the baseline calibration for the wearable. It calculates the decay rates of the molecules, which tells the wearable exactly how often it needs to sample the fluid to get an accurate picture.
So the architect designs the system and gives the wearable its marching orders. But in return, the wearable provides something the lab machine can never get, which is continuous, messy, real-world context.
Precisely. If we connect this to the bigger picture, let's look at a specific scenario from the text. Imagine a patient wearing a continuous lactate sensor. Suddenly, the wearable detects a massive fourfold spike in lactate.
Which is bad, usually.
Right? Chemically, a massive lactate spike indicates severe physical exertion or cellular oxygen deprivation. But the wearable's other sensors note that the user is completely sedentary and their heart rate is resting.
So the security guard out in the field spots a massive anomaly. The data doesn't make sense given the context.
Right. In a traditional one-way system, that anomaly triggers a false medical alarm, which causes panic and wastes clinical resources. But in a bidirectional loop, this anomaly triggers a verification protocol.
What does that protocol look like?
Well, a stored sample from the wearable's microfluidic reservoir is sent to the lab, or the user provides a fresh sample. The mass spectrometer then analyzes it with perfect, untargeted precision.
It basically calls the architect to verify what the security guard thought they saw.
Exactly. And the outcome of that verification improves the whole system. If the MS confirms that the lactate didn't actually rise, we know the wearable experienced a false alarm, maybe due to sensor biofouling or cosmetic lotion interfering with the skin.
Ah, lotion. That makes sense.
Yeah. And then the wearable's AI filter is updated to ignore that specific chemical noise profile in the future. Conversely, if the MS confirms it is a true, unexplained biochemical rise, the algorithms adapt.
So the system learns.
Right, the developers might widen the wearable's calibration range or use the MS to discover new secondary markers to add to the patch to better understand this unique metabolic event.
But to get to a point where we trust an AI patch and a lab machine communicating to monitor our health, we have to prove it's safe. The paper details a strict validation ladder.
Yes, the validation ladder is crucial.
We don't just slap an experimental prototype on a patient. It starts in vitro testing the bare sensors in controlled fluid solutions on a lab bench. Then it climbs to ex vivo testing the sensor using real human biofluids, but still safely in the lab.
Just to make sure it works with the real chemistry.
Right. Then comes short-term on-body testing, seeing how the liquid metals handle real-world temperature shifts and physical motion. And finally, it scales to independent cohorts testing across diverse human populations to ensure the sensor works universally, regardless of age, sex, or underlying health conditions.
And as we climb that ladder, this raises an important question. We fundamentally redefine what a biomarker even is. This co-adaptive structure shifts personalized healthcare from episodic diagnostics, finding out what went wrong after the fact, to predictive, context-aware monitoring.
It changes the whole definition.
It really does. A biomarker is no longer a static number on a laboratory printout. It becomes a dynamic trajectory. We are suddenly able to measure the angle of a hormone's rise, the specific speed of its decay, and how perfectly it synchronizes with your personal circadian rhythms.
So what does this all mean? It's a complete paradigm shift. We started this deep dive looking at a blurry, delayed photograph of human health. We've explored how relying on painful, delayed blood draws is slowly becoming a thing of the past.
Thankfully.
By tapping into the literal microscopic leaks of our biology, our sweat, our tears, our breath, and using the monumental magnetic weighing power of mass spectrometry to discover what those fluids mean, we built the foundation.
Yep, the discovery engine.
Then, by embedding those discoveries into soft, stretchable, liquid metal wearables, we created a continuous biological feedback loop. We are finally turning that blurry photograph into a high-definition movie.
It is arguably one of the most important shifts in medical science. We are transitioning from asking the body, what is wrong with you, long after an illness has taken hold, to continuously asking, how are you responding to the world in real time?
And that leaves us with a final thought for you to mull over. If your sweat and tears are constantly broadcasting your exact biological state, and a smart bandage can detect that your cellular energy is dropping and an infection is brewing before you even feel your first symptom, how will that change your relationship with your own body?
It's a deep question.
Will you feel more empowered and in control, having all that predictive data at your fingertips? Or will knowing every single microscopic fluctuation change what it means to simply feel healthy? Thank you for joining us on this deep dive into the future of diagnostics. Stay curious, and we'll catch you next time.